I would like to introduce Dr. Huizing. Marjan, welcome to the talk. We are going to let you take over the reins and take it from here.
Okay, thank you, Lale, and welcome everyone. It's my pleasure to present today about our NIH research studies for GNE myopathy. And as Lale said, I will present three topics of our studies: genetics-related studies, biomarker studies, and about an update and short history about our ManNAc therapy-related studies. So I'll start with the genetic screening and diagnosis for GNE myopathy. The GNE myopathy disorder is an autosomal recessive inherited disease. It means that parents are unaffected but carriers of one mutation on one of their chromosomes, and they can give this to their children. If each child receives one mutated chromosome from their parents, or so-called allele, they will develop GNE myopathy, autosomal recessive inherited. It's a young adult onset disease, starting usually with foot drop, progressing to progressive muscle weakness and atrophy. And only a little into the disease, a relative sparing of the quadriceps muscle becomes apparent. Now, these symptoms, as well as the autosomal recessive inheritance, is not extremely specific for GNE myopathy. There are several other neurological disorders, muscle disorders, that have similar symptoms and inheritance. So many patients may get misdiagnosed at initial symptoms. There's also an unfamiliarity of providers, of neurologists with GNE myopathy. And often at start of symptoms, a muscle biopsy is taken. Specific for GNE myopathy are the so-called rimmed vacuoles that you see here, that you see here on the muscle biopsy of a patient, and some smaller atrophied fibers. However, this is also not specific for GNE myopathy. There are other muscle disorders that have similar histology, muscle histology indicators for diagnosis. Unfortunately, there is not a very specific test that can diagnose GNE myopathy on muscle biopsy. And also, if a muscle biopsy is taken at an early stage in a patient, it can be taken from an area of muscle that is not very affected yet, and the rimmed vacuoles are not present in such a biopsy. So even after taking a muscle biopsy, the diagnosis can be missed. What was also confusing to doctors and patients and did not help the diagnosis is that there were so many different names for this historically for this disorder, including hereditary inclusion body myopathy, HIBM, distal myopathy with rimmed vacuoles, DMRV, mostly called like this in Asia, inclusion body myopathy type 2, quadriceps sparing inclusion body myopathy, or Nonaka myopathy. In 2014, we wrote a manuscript together with all GNE myopathy research groups in the world and renamed the disorder to the umbrella name GNE myopathy, and it is pretty well established now, the name GNE myopathy for this disease. The disease is caused by mutations in GNE, established in 2001. And the ultimate diagnosis for GNE myopathy is the presence of 2 mutations in the GNE gene. That ultimately confirms the diagnosis. This is important, and I'll get back to this later. The fact that the GNE gene was found as a cause of this disease opened prospects to identify disease mechanism and to work on possible therapies. Here is a slide from Dr. Carrillo summarizing 44 patients that she had seen in the natural history study at NIH. And it's a good example of how common misdiagnosis or delayed diagnosis is in GNE myopathy. So 35 of the 44 patients, which is 80% that she enrolled, were misdiagnosed at showing of initial symptoms. Mostly misdiagnosed with limb-girdle muscular dystrophies, Charcot-Marie-Tooth, or other muscular dystrophies. This is very common, and these disorders is very common, this kind of misdiagnosis, and these other disorders mimic the onset, in their onset, also the onset of GNE myopathy. In these 44 patients, the mean diagnostic delay was about 10 years. That means that they got ultimately the diagnosis of GNE myopathy 10 years after the onset of symptoms. This is unfortunate because in those 10 years, they could have progressed to a more severe disease, and they could have lost a window for therapeutic options. The range of the delayed diagnosis was between 0 and 32 years. A little bit about the GNE gene. Again, as I said, biallelic, 2 mutations in the GNE gene cause GNE myopathy. The GNE gene encodes or can be translated into in humans into 2 main isoforms, the so-called GNE1 and the GNE1 and the GNE2 isoform. When the GNE gene was identified, there was only the GNE1 protein known, and many patients who had been genetically tested since between 2001 and 2014 have mutation results that is according to the GNE1 so-called nomenclature. In 2011, another so-called isoform of the GNE protein was described, and this isoform is 31 amino acids longer than but otherwise completely similar than the previous GNE1 isoform. And mutation naming nomenclature rules are that you have to name mutations according to the longest available protein isoform. So that means that all GNE mutations have to be named according to the new GNE2 isoform nomenclature. That means that for most patients that had genetic testing done before 2014, their genetic reports will report the mutations 31 amino acids shorter than on the new nomenclature. Most geneticists and most scientists or doctors that know about GNE myopathy know this nomenclature change. However, new people in the field, new geneticists may sometimes mention that that a patient was that the mutations were not correct. So this means for the more common mutations in GNE myopathy, like the Middle Eastern mutation, was previously called M712T, and that is what some patients may have in their mutation report. It is now M743T. The common Japanese mutation changed, and the common Indian mutation changed from V696M to V727M. There are at this point about 260 GNE mutations reported in the literature associated with GNE myopathy. These mutations are located all over the GNE protein. And here are a few more common mutations indicated. That includes M743T, the Middle Eastern mutation, the Indian mutation V727M, and several others. So we are currently preparing a manuscript to publish with all mutations reported, which is an update from our 2014 paper. But we also include all reported patients in the literature with their biallelic mutations. This document will be very helpful for pharma, for FDA, for initiation of clinical trials to know how many patients are identified, and also will be helpful for genetic testing to see if new mutations identified have already been found in other patients previously. One interesting aspect of GNE mutations is that all patients have at least one missense mutation on one allele. And the other allele is usually also a missense mutation. A missense mutation is that one amino acid changes to another. There are no truncations or large deletions in the protein. Those are called different, like frameshift, nonsense, indel, or these other terms. And these mutations often cause in truncation or no expression at all of the GNE protein. So this means that all patients with GNE myopathy have expression of the protein due to at least one missense mutation. And there are no patients that have no protein present. So the protein that is there has decreased activity and function, but it is there. Okay, so and then, yeah, as I said, an accurate diagnosis, which is based on biallelic mutations in GNE, is very important to enroll in clinical trials, because you do not want patients in a clinical trial that have GNE myopathy-like symptoms. You really want to have patients with genetically confirmed GNE mutations in your trial. So how does this work, the mutation analysis? So in the US, it's so-called CLIA certification of GNE mutations that confirm the diagnosis. CLIA is short for Clinical Laboratory Improvement Amendments, which is a regulation that sets standards for laboratory testing of human tissues for the purpose of diagnosis, prevention, and treatment of disease. So it ensures the validity of the mutations. So to enroll in NIH clinical trials for GNE myopathy, a CLIA report confirming 2 mutations in GNE is required. If patients present a research report or an email listing their GNE mutations, it is not sufficient for clinical trial enrollment. And a doctor can order a CLIA certification, and insurance can be billed. In most cases, they pay. In our NIH trials, we can also help with CLIA certification or sending patients, helping patients to contact a certified laboratory. There are several, actually many, CLIA genetic testing laboratories in the US that can certify these mutations. So some are, for example, PerkinElmer Genomics, Prevention Genetics, Invitae, EGL, GeneDx. There are several of such laboratories. So that ends my part of the diagnosis and GNE mutations. Then I'll discuss biomarker discovery. A biomarker, as you heard from Dr. Kelly Crow last week, is a parameter that measures the presence or progression of a disease. So biomarkers can inform diagnosis and response to therapy. They're important to be identified for clinical trial purpose. So to understand the biomarkers that I will talk about, I need to first discuss the mechanism of GNE myopathy, which involved sialic acid biosynthesis and sialic acid metabolism. Sialic acid is a monosaccharide, so which is a small sugar that is present at the end of proteins that have a so-called glycoconjugate chain on it. The glycoconjugate chain is made of different sugars, different monosaccharides linked up in a chain. And then sialic acid is always the final amino acid that caps off such a glycoconjugate. Sialic acid has a negative charge, and it looks like this in membranes. Here is the protein, here's a glycoconjugate chain, and the sialic acid sticking outside of the membrane. And this is where they are very involved in signaling, signal transduction, those kind of functions. So you can imagine in muscle cells and muscle cell membranes that undergo a lot of contraction and relaxation, that these sialic acid groups are important for the cell-cell interactions. So how do these proteins get the sialic acids? Well, this starts inside the cell with glucose, which is converted into several, it can be converted in different pathways, but one pathway is it becomes a UDP GNEC, which is also a sugar. It's a charged sugar. Which this UDP GNEC can be converted to ManNAc and directly to ManNAc 6-phosphate by the GNE protein. The GNE protein has two enzymatic domains. One is called epimerase enzymatic activity that converts UDP GNEC to ManNAc, and the other is called ManNAc kinase activity that converts ManNAc to ManNAc 6-phosphate. Then two other enzymatic steps will make sialic acid. As you see, it all happens inside the cell and the so-called cytoplasm. Sialic acid is negatively charged. Sialic acid is taken up in the nucleus where it becomes charged with a CMP group. It's an energy group. It goes out of the nucleus, and this CMP sialic acid can be put onto proteins that are made in the Golgi complex. And these proteins leave the Golgi complex completely charged with sialic acids and can usually get inserted in membranes where the sialic acids stick out and can perform their function. CMP sialic acid can also feedback inhibit, that means stop, GNE enzyme to prevent it from making more sialic acid. Sialic acid, because of its negative charge, it's likely that the cell cannot endure too much negatively charged sialic acid in the cell. So this is how it shuts this cycle down. And if more proteins are needed to be made, then more CMP sialic acid can go to the Golgi and make more proteins. So what happens if you have mutations in either one of these two enzymes of GNE activity? So you do not, remember I said no patient has no GNE. So you always have some residual activity, you have some residual GNE protein left. So you cannot form enough sialic acid. So there is enzyme activity, but decreased. You cannot make enough sialic acid, and proteins likely do not receive enough sialic acids on that. So what we and one other aspect is, oh no, I'll get to that later. So what we tried at NIH quite early on when the GNE gene was linked to GNE myopathy was to give patient cells, or later a mouse model, this uncharged sugar monosaccharide called ManNAc. It stands for N-acetylmanosamine. ManNAc is a sugar that crosses membranes easily, and it occurs right in the cytoplasm, and that is where GNE is. So GNE can then convert ManNAc directly into sialic acids and can be used to make sialylated proteins. And note that where ManNAc comes in this pathway, it is after this feedback inhibited step. So by giving ManNAc, you can make as much or as little sialic acid as you want by changing the dose. Another fact is that if you have mutations or a not very well working ManNAc kinase enzyme, because of mutations in this kinase part of GNE, you can still make enough ManNAc 6-phosphate and sialic acid, because there's another enzyme in the cell, so-called glucnac kinase, that can take over. And it's been shown in the literature that glucnac kinase can convert ManNAc to ManNAc 6-phosphate. Okay. So there are other therapies that have been showed to have efficiency in cells and in animal models. One of them is sialic acid. So you could give cells, mice, patients, sialic acid itself. However, sialic acid is has a negative charge and has an entry into cells that is a different path, goes through a different pathway than ManNAc, because of its negative charge. So what happens with sialic acid, it gets encapsulated by membranes, so-called vesicles. It gets sent in small vesicles to the lysosomes of the cell, and the lysosome sorts this free sialic acid and exports it from the lysosomal membrane into the cytoplasm. So there is a more complex pathway involved to get free sialic acid inside the cell. So it may be a little less efficient in how it feeds into this pathway and increases sialylation. Some other therapies have been tested in animal models and in cells, and actually in the IVIG case in patients. You could give highly sialylated proteins that include sialylactose or intravenous immunoglobulins, IVIG. The cells will break these proteins down, and the sialic acid groups that free up can potentially be used to reuse to sialylate the proteins that need it more. And an ultimate therapy, maybe gene therapy, that is bringing a full gene, a full corrected gene into cells. So in addition to the mutated protein, these cells will make a normal GNE functioning protein, and sialic acid can be made through that. Gene therapy strategies are being worked on for many different diseases now, and some have come into clinical trials, and there is an initiative now to do preclinical research to get GNE gene therapy going. And we at NIH are a little involved with this, and we can help with the preclinical studies. So one more slide about ManNAc. Why ManNAc? Why did we not pursue sialic acid or gene therapy or sialylactose when we at this point? Why do we put our efforts into ManNAc? Well, we think it's a very efficient and safe drug, because it's uncharged. It has easy entry into cells. It has historically, by biochemists, commonly been used in cell cultures to increase sialylation. It was therapeutic in cell and mouse models of GNE myopathy, not only our models, but also of other investigators, other groups. And it's now being used in other models, rat and mouse models of other diseases that show hyposialylation. It's shown safe in mice and in humans through our phase 1 and phase 2 studies that I will get to a little later on. And it is a simple oral therapy. So it's very easy to adjust the dosing. You can just stop taking it and you will not make more sialic acid. So you can adjust the dosing according to the amount of sialic acid needed. It's not permanent. Okay, then I get finally to the biomarkers. So what would be good biomarkers? So we first looked at good biomarkers for the therapy that we were studying, ManNAc. So we for our therapy, the amount of free sialic acid is a very good biomarker. The amount of free sialic acid inside cells, but also the amount of free sialic acid in plasma. Because if we give ManNAc, free sialic acid can only be made inside the cell. So if we give ManNAc and we can measure an increase of free sialic acid in the blood, it means that this ManNAc has seen a cell. So that is a very good marker that your therapy is inserted into cells. If you would do sialic acid therapy, measuring free sialic acid in blood is not such a good biomarker, because you do not know if this sialic acid has seen a cell. Another good marker that we identified is CMP sialic acid inside cells, because this is the ultimate so-called precursor to add sialic acid onto proteins. CMP sialic acid is a little bit decreased in GNE myopathy patient cells. And after ManNAc therapy, we see it increasing. Another good marker, of course, would be to measure sialylation on specific proteins, and especially on muscle membranes. So we pursued that too. And then there are other methods. You can do large screens for certain metabolites that are different in GNE myopathy, different gene expression patterns, or other larger data sets to see if you see a trend that changes in GNE myopathy compared to normal. And we have been working on this as well. So I like to focus a little bit about the muscle membrane biomarkers. And Dr. Crow talked about this last week also, that she is investigating this in cells. So a muscle membrane is shown here with proteins inserted, and these little red things are glycoconjugates with potentially kept off by sialic acid groups. Then there are these so-called lectins that Dr. Groh talked about. Those are molecules that recognize end groups of these glycoconjugate chains. So the lectin SNA binds to sialic acid attached to a protein. And we can stain muscle biopsy slides with this lectin SNA with a fluorescent marker on it. And then we can look in the microscope where this fluorescent shows up. There's another lectin that's called VVA that binds only if sialic acid is not present. And of course, you can do a similar experiment, label that fluorescent and see where it shows up under the microscope. So here's an example of SNA staining in a muscle biopsy of a control of a normal muscle. And you see very bright green membranes, meaning a lot of SNA, meaning full sialylation. While in a patient with GNE myopathy biopsy, you see that this SNA staining is greatly reduced in the muscle membrane, indicating desialylation. The same thing is shown here, the control, good SNA signal, and in the patient's lesser signal. And here is VVA is shown here too, which is absent in, as good as absent in control muscle, indicating full sialylation, while patients show staining of this VVA, indicating absence of sialic acid. We have published this several times. So then we have also used these lectins in our mouse models of GNE myopathy. A mouse model of GNE myopathy shows the same lectin staining pattern as humans. So full sialylation in control mice and absent SNA signal in mutant mice. The same for VVA. In the normal, it's almost absent, and in the mutant, you have a strong VVA signal. after ManNAc therapy, in this case 12 weeks, in drinking water of these mice, you see the SNA signal fully coming back. So ManNAc in drinking water resialylates muscle membranes, while the VVA signal is greatly decreased. Another mouse model of this disease, studied by a Japanese group, also gave ManNAc to a different mouse model of GNE myopathy. This model greatly mimicked the human disease. The histology was similar. This was in Dr. Nishino's group, and they tested muscle contractional muscle performance, mostly muscle related strength, in these mice after ManNAc therapy, and they saw it increased after therapy compared to no therapy, indicating that the resialylation that we see with the lectins in these mice likely also occurred in the Japanese mice, although they did not do lectin staining on these mice, but they saw the increase in strength. So these are very strong preclinical data for ManNAc therapy. So we wanted to use this biomarker in our phase 2 study for ManNAc in patients with GNE myopathy. However, to use such a marker, you need to validate it. You need to show that an independent investigator that can perform this test and can an unbiased investigator and can get similar results. So we worked on this for a while. And published the validation of this test. So we developed a method to co-stain the lectin SNA with the muscle membrane marker caveolin, labeled one green and one red, and then image the entire muscle biopsy slide in our case, about 100 overlapping little so-called tiles by a microscope that can be done automated. And then these data can be sent, we sent them to a company that helped us develop software to quantify how much of the green signal co-localized with the red signal, so that how much SNA is in the same spot as caveolin in control muscle compared to patient muscle. So you can quantify the signal in the entire biopsy. You can count every single cell. And we can do a similar method of patients before therapy and their same muscle biopsied after therapy to see if we can see an increase in the same muscle of the same patient, which we did in the phase 2 study. So in the validation process of this method, we showed that GNE myopathy muscle membranes are about 50% desialylated compared to control muscle. So it's not that the sialic acid is absent, completely absent, but it's desialylated. So, and as I said, this method was then implemented in the phase 2 study. So this could potentially be the biomarker for GNE myopathy. However, we need a muscle biopsy for this, which is quite invasive. And if you're going through a clinical trial, you may want to take multiple muscle biopsies to see if your therapy is working, which may be too invasive for a patient with a muscle wasting disease to take muscle biopsies. So it is better to have a different biomarker that is easy accessible, like a blood marker or a different tissue marker. So we wanted to look inside cells, as I said, to look if CMP sialic acid and free sialic acid increased in patient cells. And to mimic a cell, we extracted white blood cells, which are whole cells, and measured ManNAc, free sialic acid, and CMP sialic acid inside white blood cells. It's easy accessible. It's a cell system. We developed a validated method with Alliance Pharma, who has this method running now. And we published this just recently, and it was used as a biomarker also for our phase 2 trial. In the past, we have also looked at biomarkers for in plasma using so-called glycomics studies, glycan analysis, glycoconjugate analysis, and we found that the amount of the so-called T antigen versus the sialylated T antigen was increased in patients with GNE myopathy. There was more so-called T antigen circulating in blood. We published this, and the method is quite cumbersome, and we need to revisit this to try to find an easier accessible method to further develop this method to be used as a valid biomarker. We are now also working on so-called 2D overlay gels to find a good blood biomarker for GNE myopathy. So that is kind of explained here how that works. So we took, in this case, blood from 6 control individuals, and we colored all their sialylate or all their glycoproteins green. So we put a green fluorescent label on all these proteins. And we pulled, in this case, blood of 3 patients with GNE myopathy, and we colored that red. And then we mixed the blood. And then we ran this mixture onto a gel. First, we separated it by charge. Remember, sialic acid is negatively charged. So if you have less charge, it gets less pulled to one side than if you have more charge. And then we separated it by size. So then you get all these little different dots. They represent proteins, glycoproteins in your blood. And then we looked what the color of these under fluorescent detector. We saw what color these dots were. If they're yellow, it means there's an equal amount of in control as in GNE myopathy, because green and red forms yellow dots. But if you see a green dot, like enlarged, like here, this seems to be a protein that is only present in controls, but not in patients, because we do not see any yellow here. And here is a little dot. It seems to be only present in patients and not as much in controls because it's more red. So then there is a machine that can pick these spots, which we did, which we performed, and can then, by mass spectrometry, analyze what the protein is. So we have this whole list of proteins now, and we know which are different in controls and which are different in patients. And the analysis process is ongoing. We did a similar experiment of patients from our phase 2 trial before therapy in green, then, and after therapy in red, and see how the ManNAc therapy changed proteins in patients. And then we can see if these proteins are similar as the ones that are different in controls and versus patients, and then you can identify a possible valid biomarker. We're studying other exploratory markers that came up in the literature as well. This is a part where NDF provided us with some funding for, and Dr. Leuklang is performing these studies in our laboratory, and we're grateful to NDF that we can continue performing these experiments. Okay, then the last part, clinical trials for GNE myopathy. I'll go quickly over this because David Clements, who will talk in 3 weeks, will provide a more accurate, or a more detailed, give more detailed information. So the clinical trials, clinical studies at NIH, they're performed by Dr. Carrillo as a principal investigator. Our boss, Dr. Gall, is the IND holder and was clinical director of the Human Genome Research Institute. And Dr. May Lectin and myself are staff scientists involved in these projects. The studies are mostly performed at NHGRI, but when we started out, the so-called Therapies for Rare and Neglected Diseases program out of the NCATS Institute helped us to get through preclinical research and get a phase 1 study started. So since the gene was identified for GNE myopathy in 2001, we have been performing preclinical research, because from very early on, we were thinking that ManNAc could be a good therapy for this disorder. A natural history study was started by Dr. Carrillo in 2011 to establish clinical trial endpoints, just to learn more about the progression of the disease, to learn more about certain markers of disease. The phase 1 study for ManNAc was finalized in 2013 and established safety, tolerability, and how the body processes ManNAc. It's called pharmacokinetics. A phase 2 study was finalized in 2018. It's for more safety of ManNAc, biomarker testing, and testing of potential clinical endpoints. We are still finalizing the final data of this. A little bit more about this later. And we're getting ready to start a multicenter pivotal study for ManNAc in GNE myopathy to show ultimate efficacy of the drug for FDA registration. As I said, natural history study details you can find on clinicaltrials.gov. This is the number, but you can just do a search for GNE myopathy and it will come up on clinicaltrials.gov. Started in 2011. There are, I think, more now than 54 patients enrolled. Patients come at different time points to NIH to get a full muscle strength testing and other disease profiles performed, so the progression of their disease can be mapped. As I said, is to identify endpoints for clinical trials. This is very important. It is important that you can convincingly show a treatment effect in a clinical study. So that means the ability to interpret that the effect of your therapy is also meaningful, clinically meaningful for patients, in that it can improve their daily activities, or it makes the patients feel better and feel stronger. So you need a very specific outcome for a clinical trial. And unfortunately, in rare diseases, it has been difficult to identify well-suited endpoints, and some trials have to be stopped because the endpoints are not well chosen or identified before the trial starts. So the natural history study is also used to identify biomarkers, both for diagnosis and drug response. I talked about that a little bit. A so-called disease progression model was developed specifically for GNE myopathy. It was published last year, and this disease progression model will be used as one of the outcomes for the upcoming trial. And patient-reported outcomes, so that is, those are questionnaires for patients to identify if patients can improve on certain therapies. So it is important to create a patient-reported outcome that is very specific for this disease, and can show and define questions that can show improvement after therapy. Some of these data were published, and a new patient-reported outcome for GNE myopathy has been created and will be used in our upcoming trial. So our phase 1 study, is to show safety and PK, how the body handles ManNAc. It's finalized, 22 patients. We developed a so-called validated method for ManNAc and sialic acid in blood, so an independent investigator can analyze this. We published that. So the trial showed that ManNAc was safe and well tolerated in a single dose. No serious adverse events after a single dose. Importantly, we saw if we gave a single dose of ManNAc, free sialic acid was produced. We could identify free sialic acid in plasma, which is shown here. I'm sure you have seen these graphs before, and it's better explained in this publication. So if you take a single dose of ManNAc, ManNAc peaks after 2 to 3 hours in your blood. It's indicated here in black. This is if the control levels of ManNAc in patients that took the placebo after a single dose of 3 grams, after a single dose of 6 grams, and after a single dose of 10 grams of ManNAc. And you see that in all doses, after about 12 hours, ManNAc disappears from plasma. This supports twice daily dosing of ManNAc. So it disappears and you get a new dose and a new dose and a new dose. Importantly, after taking a single dose of ManNAc, the free sialic acid levels in plasma start to increase after 3 grams of ManNAc, 6 grams of ManNAc, and 10 grams of ManNAc. And they peak around 10 hours after taking a single dose. And importantly, after 48 hours, after taking one single dose of ManNAc, the free sialic acid in blood is still increased, indicating that ManNAc is a slow release sialic acid therapeutic. So based on the phase 1 results, a phase 2 study was designed, 12 patients. So more information can again be found on clinicaltrials.gov, including outcomes. Patients were dosed 6 grams twice daily for 30 months. Clinical and biochemical parameters, including biomarkers, were assessed. That included strength, MRI, patient-reported outcomes, plasma and intracellular markers, and muscle biopsies were taken at baseline and about 90 days of ManNAc therapy. The objectives was really to establish safety and tolerability, and PK, this is how the body handles the ManNAc, and PD, pharmacodynamics, early efficacy, that if the drug works. And the secondary outcomes were to identify a good PRO, if the patient-reported outcome, and other efficient clinical endpoints for subsequent trials. So these endpoints can be used for a ManNAc next trial, but can also potentially be used for other therapies for GNE myopathy. So this is not only an NIH ManNAc study, but these outcomes can be used for a potential, let's say, gene therapy trial for GNE myopathy. So the outcomes, there were no serious adverse events in patients that were taking ManNAc twice daily for 30 months. However, there were some GI events reported, and it's likely due to unabsorbed ManNAc. And the tolerability became better if patients were given ManNAc 3 times daily instead of twice daily at a lower dose. And that will be performed in our next trial. Okay, I will skip this. So the next trial is a multicenter trial of ManNAc for GNE myopathy. Most of you have heard of this. It is on clinicaltrials.gov where you can find the information. It is sponsored by NIH through a UO1 grant, the NIAMS Institute. And it's in collaboration with the NeuroNext network, which is a network for excellence in neuroscience clinical trials. And some of the hospitals mentioned here are the centers mentioned here are NeuroNext sites. The PIs will be Dr. Carrillo and Dr. Amato. And our pharmaceutical company collaborator is Lydiant Biosciences. So this trial was planned to start enrollment in the spring of this year. However, due to the COVID pandemic, it was not safe to admit patients or to enroll patients to this study. And the enrollment has been delayed. And there is no specific estimate for when the sites will open up at this point. And I hope that David Clemens in 3 weeks can give a little more information about enrollment for this trial. And you can find all the information on clinicaltrials.gov as well. So with that, I end this presentation. There are many institutes, people, companies in and outside of NIH that contributed to these studies. Within NHGRI, have some the most important or the most involved investigators listed here. NINDS is involved, the TREND program, the NIH Clinical Center, Rehab Medicine, of course, Lydian Biosciences, our pharma partner, several so-called CROs that helped us with different aspects of the clinical trials, New Zealand Pharmaceuticals, of course, the Neuromuscular Disease Foundation, helping us with the preclinical research and funding, and of course, the patients and families without whom we could not perform these studies and could be so far in the development of a therapy for this disease. So in case you have not met us at meetings, so this is Dr. Gall, who is our the boss of our section and was the clinical director of the institute when the GNE myopathy trials were ongoing. Dr. Carrillo, she's the PI of all the GNE myopathy clinical studies at NIH. Dr. Lectin and myself are the staff scientists, both part-time involved in part-time studying GNE myopathy. And Dr. Loy Klang, who is a PhD biologist studying GNE myopathy in the lab full-time. So then I will open it up for questions and answers. And I believe we keep this slide up during the questions and answers, so you can read the key takeaways of this talk. Thank you.
Dr. Marjan, that was an incredibly informative and very well received discussion about so many important topics. I have asked Angela Lack, who is having some trouble with her eyes and is wearing sunglasses because she's cool and is having trouble with her eyes. I've asked her to join us to moderate some of the questions, but while we get ready to do that, because there's a ton of questions that Angela's going to, sort out for you and read, I wanted you to please clarify one thing for our audience that is a typical question, and you sort of touched on it, but the difference between a registry, which is what NDF has, is a listing of all of its patients, versus the clear certification required for the kind of registry and natural history registry that you guys need. Could you speak briefly to that, and then I will let Angela take over from there.
Yeah, sure. So yeah, a registry is more a listing of patients or a grouping of patients or a database of patients with as much or as little information this registry chooses to add. Like you can add medical records, or you can only have contact information of each patient. Registries are important for starting clinical trials, for companies who want to start a clinical trial in a certain disease, to have access to patients and to get a good idea how to contact patients and how many patients would be interested in their disease. So this is actually what we are doing with the document that we are preparing, that is going to list all reported patients so far. So it is another publicly available document, similar to a registry. A natural history study is a study, it's an IRB approved clinical protocol where patients enroll and usually come in, it's not necessary for some natural history studies, where that have very specific goals or endpoints, like they want to map the progression of muscle strength in GNE myopathy, and for that patients need to come in to get their muscle strength tested. I think your question is also related to this CLIA certification, which is not necessarily needed for a natural history study, but it is needed for a treatment study, because you really need to make sure you're treating patients with GNE myopathy,
right?
To identify efficacy of your if your therapy is working. And the only confirmation of having GNE myopathy at this point is having 2 mutations in the GNE gene, and this should be identified by an independent genetic testing laboratory, and that is CLIA certification. Now, if you do not have CLIA certification, you can still contact clinical trial for enrollment, and they will give you guidance on how to get the CLIA certification performed. It's not that then you're not eligible.
Right.
They will work with you to get this performed.
And so then to clarify for the patients listening in, because we do get this question, is first of all, everything that a patient shares with NDF is absolutely confidential. What we have is a registry of all of our patients that need to hear from us, and if they share and volunteer their own information, that's great. It stays that way. However, that's a very different thing than what Dr. Marjan Huizing just explained. So there is some clarification about that. So,
yeah.
But once again, all of these registries do stay confidential, and they'd be identified if and when we discuss them with scientists.
Yeah, and they are important. It's important to because not all patients in your registry will enroll or will be eligible or will be coming to the NIH natural history study. Because for the natural history study, we may not need more than 50 to 60 patients to map the progression of the disease. But there are many, many more patients, and it's very good to have these in a registry and know how to contact them as a group.
Right, and so for our purposes, it's so that we can be here to support you, the patients, with all kinds of efforts that the community is undertaking, not just the clinical trials, which is a very, very different thing. So I just wanted to underscore the difference between the need for these registries. One is so that we're here to support you and to make sure that you've been properly diagnosed and so on and so forth, because as Marjan points out, in rare disease, there is a lot of misdiagnoses in both directions, away from us, and patients that are referred to us as GNE myopathy patients that are actually not in fact GNE myopathy patients. So that as we try to clean this up, it is really important that those of you who want to volunteer your own information, that you do so that we can support you. That's our only purpose, very different than what the clinicians do. So thank you again for underscoring that very important topic. And I'm going to turn it over to Angela, and I'm going to disappear for another meeting. But Angela, thank you so much for pinch hitting and fielding the questions.
Not a problem. Thank you, Lali, for your introduction, and thank you, Marjan, for that very insightful talk in which you covered 3 really important takeaway areas in the field. So just to quickly touch upon these again, the first is the genetics of GNE myopathy, where Marjan spoke about the different mutation types, the importance of having a unifying naming system that's standardized amongst all the laboratories all over the world, and also the need for having a clear certification of your mutation in order to be eligible for clinical trials. So this is akin to, say, having your car being having the emissions, going to a certified place to do an emissions testing with a proper documentation that you can then take to the DMV. It's something like that.
Yeah.
So the second is biomarker discovery. So this is where scientists take advantage of what's known about the function of GNE, which has been established due to many years of research, and to then find out what are some important biomarkers that can distinguish between disease and non-disease tissue, and why this is important for diagnosis and also to guide in therapy development. And lastly, there was a brief description of the ManNAc trials as well as the natural history study, why this is important. And this is really to establish, as Marjan says, the endpoints for the clinical trials and how we will know if a therapy is working or not. That is what a natural history study is for. So, sorry. And so lastly, I just wanted to point out that the natural history and the biomarker studies, once these studies are completed, they will be applicable to all therapy development that are being established, and it doesn't need to be redone. And both these two key things, natural history and biomarker studies, they are really important in rare disease research, and you'll frequently find that a lot of rare disease foundations will dedicate a lot of resources to establishing these two types of studies. So let me just have a look at the chat history and read out some of the questions that were posed to you, Marjan, during your talk. I believe Atul has a question. He says, I have one common mutation and a large deletion in the GNE gene. Does it confirm the GNE myopathy case? And did you have something to say about that? And he also says something again at the bottom that says his muscle biopsies he's been told confirms GNE myopathy, but in my genetic report I have one common mutation and a large deletion.
Yeah, well, that is a very common question, and it is a combination of mutations that occurs often. Missense, as I said, every patient has a missense, and if it's a common missense, you know, it is, you know, that it's likely associated with GNE myopathy. Your other allele is a large deletion, which likely will not make any protein, as I showed on that slide. So it is a good start to have that, and a muscle biopsy confirmation where likely rimmed vacuoles and atrophic fibers were found. And that can go to a CLIA certified lab to confirm this if it is not already done, which your mutations may have been identified in a CLIA certified laboratory, and they wrote a letter that these two mutations are disease causing for GNE myopathy and the reasons why, some of them that I just mentioned. So that is your diagnosis, such a letter of a CLIA certified lab. So it depends where this mutation analysis have been performed.
So for example, you know, we can we can find your mutations in your GNE gene, and so can Marjan. But having said that, we are not a CLIA certified lab. Is that right, Marjan? You're not a CLIA
right.
certified
Yeah, correct.
lab.
Yeah.
So you have to find out if I mean, you do have GNE myopathy. It's just that the CLIA certification is going to be important for the clinical trials.
Right.
So I have a brief question. Do you think what happens in the cases where a patient only has one GNE mutation, one pathogenic mutation, and the other one cannot be found? Would they be eligible to enroll in a clinical trial? Just Do you know that? Do you know the answer?
No, because no, they will not be eligible to enroll in a trial because the diagnosis of GNE myopathy could not be confirmed with two mutations. So a research lab could take your case on and look for there are different ways to identify a second mutation. So that's for example, that may have happened in the patient with a large deletion because it's not commonly done in a CLIA certified lab to look for large deletions. So many patients have their mutations identified by a research lab and then can be confirmed by a CLIA certified lab. So yeah, one mutation will not confirm the diagnosis of GNE myopathy and does not make you eligible for clinical trials for GNE myopathy.
Yep, I hope that's clear to everyone. The next question is Mangesh says, are the mutations on the GNE gene or the GNE protein also?
Yeah, the mutations are in the GNE gene, but the information of the GNE gene is used to make the GNE protein. So if the information in the GNE gene has the so-called mutation, the protein will have this mutation translated into amino acids. So a protein is consists of amino acids, and they are made out of your DNA. So if there's a DNA mistake, the protein may get that mistake.
I hope that's clear to you, Mangesh. So Mangesh also has another question, a follow-up one. Are there differences, hang on a second. Are there differences in how the disease presents itself depending on whether the mutation was in the epimerase versus the kinase domain?
Yeah, that has been a question since the gene was identified for GNE myopathy. There have been several studies done for this. There was a natural history study out of Japan where the epimerase domain was identified as more severe, but there was another study where the kinase domain mutations were supposed to be more severe. In our NIH cohort, we do not see a difference between patients that have two epimerase mutations or two kinase mutations, or one in kinase, one in epimerase. There's no clear correlation that we can find. I forgot to mention that when I showed the PK results for the phase 1 trial, where the single dose of ManNAc, the peaks were shown, and then how much sialic acid was made. We did not see a difference in the amount of sialic acid created from ManNAc. In any of the patients in the phase 1 trial, it was independent of the mutation. So if you had two mutations in the kinase domain, you made a similar peak amount of sialic acid after a single dose of ManNAc than if you had two epimerase mutations. So in the few patients that were in our phase 1 trial, we did not see a difference in how ManNAc is processed mutation-wise. And in our patients, as I said, there's no apparent difference in age of onset, progression of disease, or other parameters depending on mutations.
Great. Thank you.
It's difficult for a rare disease to establish this because there are such small cohorts studied all over the world. So it's difficult to group this in a large study, but this is what we know so far.
Yep, that makes sense. And Mangesh has another follow-up question. For a single cell, how often does this process of converting glucose to sialylated glycoprotein happen in its lifetime? Is it happening constantly?
Yeah, so that is a good question. So that is called turnover of proteins. So the proteins are made in the cell, they're put on the plasma membrane, And some proteins are used a lot or can break down or need to be renewed often. So it's called, these proteins have a higher turnover rate. That means they need to be renewed more than others. There are some muscle proteins that are turned over every 8 hours. There are others that are turned over every 3 days. But this occurs relatively often in proteins. It's not that you do with one protein a lifetime. So it is hours to days that proteins are turned over to be renewed in the cells, newly glycosylated, and put back on the plasma membrane. It's a very dynamic process.
Yep, that's a very good answer, and I hope that makes sense to you, Mangesh. Tara is next. She asks, if, as we see that ManNAc is entering the cells, to what degree is it restoring and repairing the pathogenic and diseased part of the muscles? And the second part is there any long-term animal or human model which indicates that ManNAc increases strength and completely arrests the disease process?
So the first question was if ManNAc enters muscle cells. Yeah. We did we did not measure ManNAc or sialic acid inside muscle cells because we think there are better uses of the muscle tissue that we got through the biopsy. But for muscle, we tested the lectin staining. So before in the phase 2 trial, a muscle biopsy was taken before the patient started ManNAc therapy. And these muscle biopsies had a certain degree of hyposialylation, desialylation. And then we did a similar, with lectin staining, we did a similar lectin experiment on a muscle biopsy taken 90 days, after being 90 days on ManNAc twice daily, 6 grams, and tested in the same muscle the muscle sialylation, and in the overall group there was an increase in sialylation. The results are on clinicaltrials.gov, and we're prepared. And Dr. Carrillo is preparing a manuscript to publish this in the literature. So we did see that ManNAc did do something to muscle cells, that sialylation increased. At the same time, muscle strength was measured in patients in the phase 2 study and analyzed with this disease progression model. And those results will also be published and made available when the analysis has been finalized. So we think ManNAc was taken up by muscle cells and resialylated membrane proteins. We did test white blood cells of patients that were on ManNAc, and we did see an increase in intracellular sialic acid and CMP sialic acid, indicating that ManNAc does get into these cells, and increases the production of these compounds. Does that answer your question?
It was pretty clear to me, and if Tara has any follow-up questions, if we could touch base offline. And also the next question is, Tara also wants to know if the mutation manuscript you're working on, will you list novel GNE-M mutations as well?
Yeah, so novel mutations not reported that were identified at NIH, they will also be included. Yeah.
Mangesh says, is there any preliminary data on efficacy from the ManNAc phase 2 trial?
Yeah, I just mentioned that. The results that I just mentioned are on clinicaltrials.gov. go to the ManNAc phase 2 trial, and you can read them there. They're all in tables, so it's a little hard to interpret. And a publication with the results will be is being prepared by Dr. Carrillo. That is all I can say at this point. The last question was if you have a mutation and it to get it verified, if your parents need to be tested, if that would be helpful to show that it is biallelic. And my answer was it is dependent on the nature of your mutation. If your 2 mutations are common mutations in GNE myopathy, we already know or have determined that those mutations are disease causing and are so-called pathogenic. So then parental testing is not needed. Sometimes parental testing is impossible because no DNA from parents can be collected. Then family members would be informative. Sometimes those are not available either. Then there are other methods to show that your mutation is pathogenic. For example, you can test with an antibody, or you can test enzyme activity, or you can test a muscle biopsy if available. There are other criteria that can add to the pathogenicity of your mutation. So these criteria will be added to the manuscript that we are finalizing for every single mutation that has been reported for GNE myopathy. So this will help screening laboratories, or the CLIA certified laboratories, identifying if GNE myopathy mutations are pathogenic or what is needed to score them as being pathogenic.
And I guess that rolls really well into the presentation, Angela, that Moncol is going to make about the samples that we've already collected. And of course, as you know, those are going through PerkinElmer, which is a CLIA certified lab, and we will be having hopefully somebody from PerkinElmer give a talk about that as well, maybe in tandem with the conversation that we're going to have with Moncol Lek. Was that the end of the questions? I thought I heard you say last question as I came back on.
I have one additional question which you can answer briefly. I just wanted to know what is your ideal biomarker in terms of what characteristics would you want it to have?
Yeah, an ideal biomarker for GNE myopathy would be a muscle marker for showing an effect in the affected tissue. However, it is not a very practical marker because of the access to muscle, and a muscle biopsy needs to be performed. So a blood biomarker will be way more practical and maybe as informative as muscle. So in our phase 2 study, we for this reason too, we collected these muscle biopsies. So if a biomarker is identified in blood, we can go back to our muscle biopsies and see the ones that are more hyposialylated, if they show more effects in the new biomarker than the ones that were almost normal sialylated. We can see if the biomarker is less informative there. So we hope with the tissues that we have available at NIH now that a muscle biopsy is not needed for future biomarker identification. We can use the already collected tissues for many future studies, including biomarker studies, and that even for subsequent ManNAc trials, there are no muscle biopsies required.
Thank you to both of you. No sound. Okay, well, thank you to both of you for your presentation. We're gonna say goodbye to everyone until next time. Thank you. Bye-bye.
Thank you.